Good morning, everyone, and welcome. I'm delighted to have you here today. Today, we'll be exploring the management of media used for microbiology analysis. Our focus will be on understanding the different types of media, their preparation, and their role in microbiological investigations. So, let's get started and see how these media help us in identifying and studying microorganisms effectively. Let's talk about why proper media management really matters in our work. First, it supports accurate microbial detection and enumeration, which is crucial for reliable results. When media is prepared and handled properly, we can trust that our microbial counts are correct. It also ensures reproducibility and reliability, meaning that our tests can be repeated with consistent outcomes, which is key for quality control. Another important aspect is compliance with regulatory standards. For example, standards like ISO IEC 17025, CLSI guidelines, and Good Laboratory Practice, or GLP, all emphasize proper media management. Following these standards helps us meet regulatory requirements and maintain credibility. Finally, good media management minimizes the risk of false positives or negatives, which can lead to incorrect conclusions. It also reduces contamination risks that could compromise our entire experiment. So, overall, proper media management is vital for producing accurate, reliable, and compliant microbiological data. Let's take a closer look at the regulatory framework that guides our media preparation and validation processes. First, we have the key standards and guidelines that set the foundation for quality and consistency. One of the most important is ISO IEC 17025, which specifies the requirements for media preparation and validation to ensure reliable testing results. Then there are the CLSI documents, such as M22, M38, and M100, which provide detailed performance criteria for media quality and performance. These documents help us verify that our media meet the necessary standards for accurate and reproducible results. We also need to adhere to Good Laboratory Practice Principles, or GLP, which emphasize proper documentation, traceability, and quality control throughout our processes. This ensures that every step is recorded and can be reviewed if needed. In addition to these international standards, local regulations like those from the EPA or FDA come into play, especially for specific testing laboratories. These regulations may have additional requirements to ensure compliance within particular regions or industries. The overarching objective here is to ensure that our media handling is standardized, well-documented, and thoroughly validated. This approach not only maintains high quality, but also supports the integrity and reliability of our testing processes across the board. Now, let's explore the different types of microbiological media that we commonly use in the lab. First, we have general-purpose media. These are designed to support the growth of a wide variety of microorganisms, making them very versatile. Examples include nutrient agar and tryptic soy broth, which are staples in many microbiology labs. Next, there's selective media. These are specialized to inhibit the growth of unwanted microbes while allowing the target organisms to thrive. For instance, MacConkey agar is used specifically for gram-negative bacteria, helping us isolate and identify them more easily. Then we have differential media. These are particularly useful because they allow us to distinguish between different groups of bacteria based on their reactions. Blood agar is a classic example, as it reveals hemolysis patterns that help differentiate bacterial species. Finally, there's enrichment media. These are designed to boost the growth of specific organisms, especially when they are present in low numbers. Selenite broth, for example, is used to enrich for salmonella, making it easier to detect in a sample. Understanding these categories helps us choose the right media for accurate identification and study of microorganisms, which ties directly into our overall theme of effective microbiological analysis. Let's talk about media specifications and selection considerations. When choosing a medium, the first thing to think about is its intended purpose. Are you aiming for enumeration, like counting colonies? Or are you isolating and identifying specific microorganisms? Clarifying this helps guide your choices. Next, ensure that the nutrient composition of the medium is well defined, ideally based on validated formulations. This guarantees consistency and reliability in your results. Another key point is to consider factors like pH, osmolality, and any additives you might include. These can significantly affect microbial growth and viability. Compatibility is also crucial. Make sure your medium works well with the target organism and the method you're using. For example, if you're working with urinary bacteria, CLED agar is a great choice because it prevents swarming and allows for accurate enumeration. Finally, always choose quality-assured sources for your media. Using reputable suppliers ensures that your media meet the necessary standards, which is essential for obtaining accurate and reproducible results. All these considerations help us select the most appropriate media, ultimately supporting the overall quality and reliability of our microbiological work. Let's talk about procurement and receipt, which are critical steps to ensure quality right from the start. First, it's essential to buy from approved vendors. This helps us maintain a consistent standard and reduces the risk of receiving subpar materials. When the shipment arrives, always check the certificate of analysis, or COA. This document provides important details about the product, including its quality and compliance. Next, verify key information on the COA, such as the batch or lot number, to ensure it matches our records. Also, confirm the expiry date to make sure the product is still valid and safe to use. Don't forget to check the storage conditions listed. These are vital for maintaining the integrity of the material. Once verified, assign an internal lot ID to keep track of everything efficiently. Then, enter this information into our Laboratory Information Management System, or LIMS, or update the media inventory accordingly. For example, when verifying a COA for media, we look at sterility and the correct pH range. These checks are crucial because they directly impact the reliability of our experiments and processes. Overall, following these best practices helps us maintain high quality and consistency throughout our work. Let's talk about storage and handling. First, it's important to follow the manufacturer's specified storage requirements. For example, dehydrated media typically needs to be stored at temperatures between 2 and 8 degrees Celsius. Maintaining proper temperature helps preserve the media's effectiveness. Humidity control is also crucial, as excess moisture can degrade the media over time. Organizing your shelves by media type and expiry date makes it easier to find what you need and ensures you're using the oldest supplies first. Implementing a first-in, first-out system is a great way to prevent wastage and ensure freshness. When it comes to handling, always keep packaging intact until you're ready to use the media. This helps prevent contamination. Also, try to minimize the exposure of opened packages to the environment. The less time they're open, the lower the risk of contamination. Overall, proper storage and careful handling are key to maintaining the quality and reliability of your media, which supports the integrity of your entire workflow. Let's walk through the key steps involved in preparing media, starting with the elements of the standard operating procedure. First, we need to carefully weigh each of the components to ensure accuracy. Next, we add the correct volume of water, making sure it's precise to achieve the desired concentration. Then, we adjust the pH to the target level, which is crucial for supporting microbial growth. After that, we heat the mixture to promote dissolution of the components. Once everything is dissolved, the mixture must be sterilized, typically using an autoclave, following the specific protocol to ensure safety and consistency. After sterilization, we pour the media into plates or dispense it into containers, depending on the intended use. Throughout this process, calculations are vital, making sure we have the correct weight-to-volume ratios. We also need to adjust our calculations for the batch size we're preparing, whether it's a small or large volume. As an example, let's consider preparing 1 liter of tryptic soy broth at a pH of 7.3, with a tolerance of plus or minus 0.2. This illustrates how precise and careful we must be at every step to produce reliable, reproducible media for our experiments. Remember, the accuracy in each of these steps directly impacts the quality of your media and, ultimately, your results. Let's talk about sterilization and quality control. When we sterilize, we typically use an autoclave set at 121 degrees Celsius and 15 psi of pressure. This combination is effective in eliminating all microorganisms, but it's not enough to just run the cycle. We need to verify that sterilization was successful. That's where Bowie-Dick tests and biological indicators come in. The Bowie-Dick test checks for proper air removal and steam penetration, ensuring the sterilizer is functioning correctly. Biological indicators contain spores that are resistant to sterilization. If they are killed, we know our process is effective. After sterilization, we conduct post-sterilization quality control checks. For example, we examine the clarity of the media, which should be transparent without any cloudiness or particulate matter. We also perform a pH check to ensure the media's acidity or alkalinity is within the correct range, indicating proper media preparation. Finally, we do a sterility check by incubating an uninoculated control sample at 35 to 37 degrees Celsius for about 48 hours. If no growth occurs, it confirms that the sterilization process was successful. All these steps are essential to guarantee that our sterilized materials are safe and ready for use, tying back to our overall goal of maintaining high standards in laboratory quality control. Now, let's talk about the performance verification of media, which is a crucial part of quality control. First, we conduct essential QC tests to ensure the media performs as expected. One of these is the growth promotion test, where we inoculate the media with defined QC strains, such as E. coli or Staphylococcus aureus, to confirm that they grow properly. Next, we perform selective and differential checks. These are important because they help us verify that the media can correctly inhibit or differentiate bacteria as intended. For example, with MAC conky agar, we expect lactose fermenters to produce pink colonies, which confirms the medium's ability to differentiate based on fermentation. We also check the pH and indicator performance to ensure the media's chemical properties are accurate, and the incubation performance to confirm that the media remains stable and effective throughout the incubation period. All of these steps together help us verify that the media will give reliable and consistent results in our testing processes, supporting the overall integrity of our quality control system. Let's talk about documentation and traceability. First, record-keeping requirements are essential to ensure that all steps are properly documented throughout the process. This includes batch records, which capture all the details of each production run, and preparation logs that document the setup and procedures used. Sterilization records are equally important, as they verify that all sterilization steps meet the necessary standards. QC test results provide the data confirming product quality and safety, while deviations and corrective actions document any issues that arise and how they are addressed. Traceability is a core concept here. It's about linking all media to specific batches, lots, preparers, and dates, creating a clear audit trail. Typically, this information is stored in a laboratory information management system, or LIMS, or within a broader quality system. For example, you might scan a barcode on a media bottle into the LIMS, which automatically records the past status of the QC test, ensuring quick, accurate traceability. All of these elements work together to maintain compliance, ensure quality, and facilitate effective investigation and resolution if any issues occur. Let's take a look at some common issues you might encounter and how to troubleshoot them effectively. When dealing with problems, the first step is always to identify the specific symptom or error message. This helps narrow down the root cause. Sometimes, issues stem from simple things like loose connections or incorrect settings, so double-check those basics first. If that doesn't resolve the problem, consult the troubleshooting guide or logs. These often contain valuable clues. Remember, troubleshooting is often a process of elimination, testing one potential fix at a time. Also, don't hesitate to seek support from colleagues or technical resources if needed. Connecting these troubleshooting steps to our overall goal of maintaining system reliability is key. By resolving issues quickly and accurately, we keep everything running smoothly and avoid larger disruptions down the line. Now, let's talk about biosafety considerations, which are crucial when working with biological materials. First, we need to conduct a thorough risk assessment to understand the potential hazards involved. This involves evaluating the type of organism we're handling and the associated risks. Based on that assessment, we should handle the media carefully, especially when working with different organism groups, to minimize any potential exposure or contamination. When working with open plates, it's best practice to use an appropriate biosafety cabinet, specifically a Class IIBSC, to provide a safe environment for both the researcher and the sample. Personal protective equipment is also essential. This includes gloves, a lab coat, and eye protection, to create a barrier against any accidental spills or splashes. Waste disposal is another key aspect. All waste materials should be sterilized in an autoclave before disposal, and then discarded according to biohazard waste rules. Following these biosafety steps helps ensure a safe working environment and is a fundamental part of responsible laboratory practice, connecting back to our overall goal of maintaining safety while conducting scientific work. Now, let's talk about regulatory compliance and how we ensure that our media management practices are properly audited. One key aspect is SOP adherence, which means following our standard operating procedures consistently to maintain quality and reliability. We also keep detailed QC logs, which help us track quality control activities and identify any issues early on. Traceability records are equally important. They allow us to trace every step of the process, ensuring transparency and accountability. Calibration of equipment is another critical element. By regularly calibrating our tools, we make sure measurements remain accurate over time. Additionally, comprehensive training documentation demonstrates that our team is well-trained and competent. All of these components come together to produce credible results and demonstrate our commitment to competence. This thorough approach not only keeps us compliant, but also reinforces the integrity of our work, an essential part of maintaining trust in our processes and outcomes. Let's wrap up with some key takeaways. First, media management is absolutely critical for maintaining data integrity. Proper handling ensures that the data remains accurate and reliable throughout its life cycle. Next, it's essential to follow standardized SOPs and QC protocols. These procedures help us stay consistent and prevent errors that could compromise our results. Maintaining thorough documentation, traceability, and compliance is also vital. This not only supports transparency, but also allows us to track any issues back to their source if needed. Lastly, regular training and audit preparedness are crucial. They ensure everyone stays up to date with best practices and that we're always ready for inspections or reviews. Together, these points emphasize a comprehensive approach to managing data quality and integrity, which is the backbone of our overall success in this field. For further support, please contact us. If you have any questions or need additional assistance, don't hesitate to reach out. We're here to help you succeed and ensure you have all the resources you need. Thank you so much for your time and attention today.